So thank you, Virginie. Also, thank you for the kind introduction to this very nice meeting. Unfortunately, I cannot entertain you with any anecdotes about me meeting Cyrus, because the first time I met him was, I think, in 2010 in Vienna, although of course, I knew the name since a long time from his work and his software products that we have been using quite extensively as well.
But I do remember one thing. Ajit Kamani and I were working on a problem of group sequential methods with primary and secondary endpoints, and we were just about to submit our paper, and lo and behold, he and his colleague Willie Moore had submitted a paper on the identical topic.
That's true.
And they
That
spooked
was
us. However, our paper was published as well.
yeah. Okay.
And then in Paris, when we had our first user group meeting, I got a better idea of his work because two of his students, I think, presented some problems that they had been working on with you on that topic, and we were very excited by that. There was lots of questions, and when we felt that when we had the next meeting, it was my pleasure to invite you.
Yeah, and thanks a lot. So it's my pleasure to be here. My talk will be along the lines of what Chris and Sue have talked about. So I will start with an introduction which might look familiar to you, things that you might have heard earlier today as well as yesterday, so I can be very quick about that regarding combination p-value methods and conditional error functions. But then I hope I can add a little bit of our recent thinking to this regarding what I've termed here in inverted comma unconventional combination methods, where we go to some extent beyond the methods that are available. And I will give two examples of that. One of them is on treatment arm selection and one is on sample size re-estimation. So quickly go over the introduction again. To me, an adaptive design is a clinical trial which is split into several stages, and there is an interim analysis after every stage, and then the subsequent trial stages that you do when you go on depend on the interim analysis result. So I think, just to give you an example, I think this trial has already been mentioned. There was a trial in Novartis in COPD, which started with 4 different dosing regimens of indacaterol. Yeah. And after the interim analysis, 2 of the 4 dosing regimens were dropped. It was foreseen that there would be a dose selection in this design, but it was not foreseen how many doses would be selected, or which doses, which regimens, for example. And this trial is published in a paper on respiratory medicine. If the only option is to stop the trial for efficacy or futility, but no other things about the trial are changed, then that is called a group sequential trial. So what are the possible adaptations that we have? The sample size, of course, it is one of the things that is under much discussions. How can we change the sample size following the interim results? You might think about changing the sample size allocation to the treatment. So, for example, changing the randomization ratio if maybe you do not want to give up completely on a treatment arm or on a subpopulation, but you may see more hopes in one of the dosing regimens. Special case of that, in a sense, would of course be to completely stop a treatment arm. You could also think about adding a treatment arm, although I haven't thought about that too much, I have to admit. Population selection, if you have pre-specified a subgroup where you hope for more benefit than another, you might think about dropping that population or changing the inclusion-exclusion criteria. And in theory, of course, as we well know by now, you could redesign the entire trial. So change everything, primary endpoints, which hypothesis, and so on. What are the benefits of that? You can correct wrong assumptions, select the most promising options early. There might be external information in your trial that is important to your trial because, for example, a competitor has brought another, I don't know, multiple sclerosis drug to the market, and it's important to you to react to that in your trial. So the hope is that, you know, you are not so fixed. It's not like your trial is now running and it's going on, and nothing that happens in the outside world will ever allow you to change this running thing. This is the idea behind adaptive designs. But as you have all heard repeatedly, if you are not properly accounting for those adaptations, then you are inflating type 1 error rates if you are in the setting of a confirmatory critical trial. And therefore, what you need to do, even if in theory you might be allowed to redesign the entire trial, is in practice you need to set out the rules for early rejection in your study protocols. You have to say what methods you will be using to combine the evidence across the stages. The class of envisaged decisions after stage 1 needs to be stated. This is a bit of a vague statement, of course. It means something like you are not totally free to change everything after stage 1, say, but it doesn't also necessarily mean that you have to fix very specific rules. It means that you need to pre-specify the basic idea. Like, for example, I will do sample size re-estimation. I will not decrease the sample size, but I'm considering increasing the sample size. If I'm doing that, I will be using a combination p-value method to adjust for the type 1 error, for the family-wise error rate. And then within these frameworks, you can make your decisions, for example, based on Bayesian principles, as we have just seen are now supported by EAST. If you want to predict the success of your study at the interim from assuming some Bayesian prior on the putative effect. Right, and then of course you need to see what this entire strategy means in terms of the operating characteristics of your trial. that just reproduces a t-test. Okay, so far so good. But of course, and I mean, so I did some simulation and this method works. But of course you would like to know how well does it work. So I was wondering, what can I compare this with? So the idea that I had is the following. I say I'm using a combination p-value approach to compare it with. So it's written down here again. You have seen this formula. You would reject the hypothesis for the selected treatment if the combination function is above the normal distribution quantile, standard normal distribution quantile, for all of the intersections that contain the selected treatment. And this pi is now the p-value of a stage 1 Dunnett test. Of course, if it's the selected treatment, it's just a p-value. Now, as this method is based on separate tests in stage 1 and in stage 2, because at both stages we are, of course, comparing here the same hypothesis, but with different data, we must split the placebo group into stages as well. So I did that at random. So I said, you will see later, I assume 12 and 30, 12 observation to the interim per group, and then 30 observation per group later. So I said I split 42 placebo patients randomly in simulations, that's no big deal, in 12 plus 30 to do this. And yeah, this is pretty fresh, so what I'm a bit disappointed about is that my suggestion doesn't give better power than the combination p-value approach. So I was thinking a little bit about why that is. One of the explanations, I think, is if you assume that you know the sigma, so that you have Z tests actually, then you can work out that the suggestion that I have made and the conditional p-value combination test are identical. They don't only have the same distribution, they are point-wise identical for the elementary hypothesis about the selected treatment. And for this, the weights are optimal. For the other intersection hypothesis, the two approaches are not identical, but the set of weights is of course not optimal for either of the two methods. So that might be an explanation why my hope, I was kind of hoping I had this idea that this back and forth transformation with the t-test statistic that is going on when you do a combination p-value approach might hurt power. But in this case, it didn't do that. But anyway, I mean, if in this setting you wanted to use the combination p-value method and not ignore the fact that you have estimated the sigma, then of course it becomes a bit tricky. How would you do that? I mean, you can of course just ignore the fact that sigma is estimated, but then you have a slight type 1 error inflation. Or you could say, I estimated only at the end of the trial, but then what's the distribution of the combination? So, I mean, just to check what's going on there, I would say that the advantage of this combination that I've suggested now is that you don't have to split the placebo group into a stage 1 and a stage 2 arbitrarily, and you avoid a back and forth transformation of original test statistics. And then I did a bunch of simulations where in this simulation for the conditional p-value approach, I simply ignored the fact that the sigma is estimated from the data. And then, of course, you can see that you plug in the estimated sigma as if it were known in the type 1 error. So I did 3 arms with 12 patients in stage 1, and then 30 patients in the selected arm, and 42 patients on placebo, 100,000 simulations with an average level of 5%. And I tried various ways of modifying the sample size after the interim, smarter and less smart ones, but the observed error was always between 4.9 and 5.1% for the suggestion of combining the means directly. So that's reassuring because that's what it should give. But for the combination p-value approaches, when you ignore estimation of the sigma, it was between 5.4 and 5.6%. But as said, if I modify this and say, okay, let's forget about this problem with the sigma estimation, let's fix that to a value that I know, then I did not see any real difference in terms of the power between those two methods. What, of course, you have with both methods is a clear gain in power over a stage 2 only test. So if you would, as an alternative, say, I cannot do this, I have to do the dose finding first, and then I do another test that confirms against the comparator, then you lose something on the order of magnitude of maybe like 7%, which is quite a bit, I would say. The power is capped at 1. So what can you expect in terms of power gain when you are already at 90%? Right. So to wrap up this first example, the phi and this combination p-value approach represent slightly different conditional error function approaches in my view. So it's different ways of coming up with that. Although I haven't written them down as conditional error function approaches. You could, of course, now say, okay, if we simulate the null distribution of the test statistic anyway to get critical values, we can also do that with a conditional p-value approach when we plug in the estimate of the variance that we obtain at the end of the trial only. I haven't done that either. This test has no intrinsic power advantage over the conditional p-value approach. What I mean by that is, of course, it's unfair to compare a method that keeps the type 1 error with a method that does not keep the type 1 error. So therefore, if you see that the conditional p-value approach has more power, if you use it in a conventional way with the approach that combines t-test statistic, then that's unfair. But if you use these approaches and just fix the sigma, I don't see an advantage of this method as I had hoped. So I don't know. The question is, should this method be considered in conventional adaptive designs as well? And the other is, of course, the big question, is it acceptable to bring in the control arm in stage 2 only in a clinical trial? That might be, I don't know, a challenge. You at least have to discuss it with the health authorities, I would say. So I have another 10 minutes for my second example. Very good. So my second example is blinded sample size review. I've talked about that before, but I've given it a slightly different twist, I hope. So what is sample size review? We all know this. Re-evaluation of the sample size at an interim analysis. In an unblinded sample size review, you would disclose the patient, the treatment by patient allocation, so you would know which patient got which treatment. In a blinded sample size review, you do not know this. So you know the results, but you don't know who is in which group. And it's intuitively clear that unblinded sample size re-estimation can introduce a bias because of the selection that you are doing. Now, what about blinded sample size re-estimation? Again, for simplicity, let's talk about the normal case. So you have observations xi from a normal distribution. You want to test, I've written it down here as the one sample t-test because conceptually that's the same as the two sample case. So we want to test the null hypothesis that the mean is 0 with the t-test. We have n1 observations at interim, and then after the interim we add n2 star observations for the second stage. And then one of the methods that has been suggested is that you, in blinded sample size review, you re-evaluate this N2 star based on the total variance. So this is the variance under the null hypothesis. You can put it that way. The equivalent expression in the two sample case is the variance calculated ignoring the treatment allocation. And N2 star is a function of this and thus of the stage 1 data. But again, this is an example where we have a restriction on the way we combine the two stages, because this is the only quantity that decides about our re-estimated sample size for the second stage. So again, we are not totally free. And then Kieser and Friede say do an unmodified one sample t-test using this n as if it were fixed from the beginning. And they claim that that keeps the type 1 error. And the method is generally accepted. So if you look into the FDA draft guidance on adaptive design, they say this is a quote from that guidance. It simply says, revisions based on blinded interim evaluations of data do not introduce statistical bias. Now, this is, let's say, this is almost true. It's true, but not quite. It has not only not been shown formally that it keeps the type 1 error, you can show that it does not keep the type 1 error. But to be fair, only in very, very extreme cases. In cases where you do something like take 2 observations and take 1 other. Things like that, you know. But, I mean, yeah. And the type 1 error inflation is minute, so they may have difficulties picking it up. But formally, this is not a method that has been proven to keep the type 1 error. It's just one thing that the health authorities have decided to be acceptable. But of course, the combination p-value method keeps the type 1 error because we can always use that. But again here, we can use the restrictions to come up with something more powerful. But first of all, again, about the p-value combination and conditional error function approach. So in that case, we would have to fix the weights. There is an originally intended stage 2 sample size, which otherwise, of course, you wouldn't need. And N2 that might move it to become an N2 star. If these two things are the same, then the power is best. And you can use that in this case. But what you will see if you compare it with the unmodified t-test at the end, you lose something like, in many scenarios, like 3 to 4% of power. You might lose up to 7% of power in some scenarios that I have tried to work through by simulation. And in this case, I think it's fairly obvious. This great flexibility of the p-value combination approach, which would allow you to do all sorts of modifications of your original trial, not only ones that are based on this total variance, which is probably not such a great quantity to calculate what the stage 2 sample size should be, it's not exploited here. And we know that it's not exploited. It cannot be exploited because we haven't been unblinded. So this method is not designed specifically for this situation, and therefore it suffers from some power loss. And there's also a small power loss, at least in this case, from transforming a t-test p-value with a normal score. That's, of course, not the optimal back transformation when it comes to combining the two stages with weights later on. So here's, again, similar suggestion, direct combination of the test statistics. There is a nice twist here. Because you have 2 test statistics, one unconditional from stage 1 and conditional from stage 2, respectively. This is conditional on this quantity, on the total variance. And there is a nice theorem from generalized multivariate analysis that you can find in this book, for example, which says that if you condition on this quantity, the t distributions of the two test statistics still hold. And they are all still stochastically independent. So you have a result about the two test statistics, and in this case, this is a different twist from our previous example. This allows you to use, in this specific situation, data-dependent weights when combining the two stages. So here you can, rather than use these pre-specified weights, N1 and N1 plus N2, you can use N1 over N1 plus N2 star as the weight. And that, of course, is the real optimal weight. This is not covered, and because of this data-dependent weights, it's not covered by conventional suggestions. But it's only allowed because the dependence on the stage 1 is exclusively via this total variance. So that is the only quantity that is used to, from the stage 1 to do the sample size re-estimation. And you are explicitly using this in this situation. Then again, this is something interesting. I wasn't aware of this before I started this. The multivariate t distribution is not the joint distribution of two multivariately t distributed quantities. It's something else. So the weighted sum of two independent t random variates is not something simple. I looked up in Johnson and Kotz. I was kind of thinking that must be a t distribution again or something. No, it's not. So, but again, of course, you can easily obtain it by simulation or numerical integration. So you get the critical values. The unmodified t-test statistic is also not a special case because that cannot be written as a weighted sum of the stage 1 and the stage 2 statistic. So this is the new suggestion for doing a combination of test statistics which are not p-values and also, not just doing the final t-test at the end, ignoring that you have re-estimated the sample size. My final few slides are about the power comparisons in these situations. So here's the setup. I did 1 million runs per scenario, sample sizes from 5 to 50 in steps of 5, and then I did some modifications of the assumed treatment effect and the true treatment effect. And after stage 1, we recalculate the sample size so as to retain 80% power with the assumed treatment effect. That is the reason why these curves are going down, because the true treatment effect is kept constant, but you assume more and more of a treatment effect, so you go with lesser and lesser stage 2 sample sizes on average in this. But I mean, in this case, I really wanted to see how this suggestion behaves relative to a combination p-value approach or to an approach that does the t-test at the end without without any modification for the sample size re-estimation. And this is the graph that you get. So this is for 5 observations in stage 1. So very small number. You can see that the Fisher p-value combination loses some power. The unmodified t-test, from which we don't know whether it keeps the type 1 error or not in this situation, is a tinsy-wincy little bit better than this t-value combination. But it's very little. And if you move on to n1 equal to 30, then those two methods almost have identical power, whereas the p-value combination approach, of course, loses some power still. Right. So this summarizes my talk. what I've tried here was a bit of a test balloon. I've discussed two cases where I would say the conditional error function approach is used in a bit of an unconventional way. In the thing that combines these two cases is that in both cases, our interim decisions are restricted, but not completely pre-specified either, so this middle ground. I think with today's computing power, some of the mathematical technicalities are really no fundamental hurdle anymore. So as long as we can write down our test statistics as a sum or as some simple function of a few known random variables, so that we find a standard representation, so to speak, for the null case, then if we are there, we can, of course, always simulate critical values and that kind of thing. And that's much easier than maybe it was like 20 years ago, when it was really important to work out these null distributions. But of course, there are still some open questions regarding the operating characteristics of such suggestions. So for example, if you're using pre-specified weights, how do you set them up? When you combine z-test statistics, that might be easy. But when you combine t-test statistics, when you move through a sequence of tests for multiplicity adjustment with a closed test principle, I mean, we know that the weights are not optimal for all the intersection hypotheses, so should we have different weights? Those kinds of things, I think there is still some room to think about. And that concludes my talk.
Ekkehard, the issue, I take it, all of this research is because you're talking about very small numbers, right? Mm-hmm. I mean, if you have a clinical trial with 100 patients per arm, then you know the fact that you don't know sigma, and all of this is because you don't know sigma squared.
Yeah, there is a lot of truth in that. Yeah, if you rely on the asymptotics of your test statistics, yeah, then these problems disappear. But I mean, of course, if you rely on asymptotics, then of course there is no issue with blinded sample size re-estimation at all, because the statement you are allowed to do this z-test at the end then converts to you are allowed to do a t-test at the end, converts to you are actually allowed to do a z-test at the end. And then, of course, I mean, there is no issue at all.
Yeah.
I think it depends a bit on how many analyses you're doing. In a group sequential test with a lot of analyses, then you can have hundreds of observations and there's still a problem.
Just to say, if you do not stop at interim, if you just say, I re-estimate my sample size at interim, and I will not stop, and I will go on. And at every stage, I re-estimate my sample size, and every stage is infinitely large, and at the final analysis, I do a z-test. There is no problem,
No, you fix the maximum information. You have to think of information-based...
the Chow-Robbins procedure, which does that, it's pretty bad. With hundreds of observations, where you say I'm
not saying hundreds of observations. Sorry, go ahead.
Well, people want to get to lunch, so we probably should talk. But I was going to make one comment. What's really intriguing is to say if you're doing information monitoring, even because you're actually unblinded, you could do your sample size readjustment using the blinded estimate of variance, and you'd avoid these biases that the Chow-Robbins type approach gives, which is very odd that you've got the information, you can ignore it, and in some ways you're better off, I think.
Yeah, I agree, but I mean, okay, maybe I'm too tied to the pharmaceutical industry. The health authorities won't believe you, I guess, if you claim that you have done it only based on the blinded sample size estimate, on the blinded variance estimate. I would think. Yeah.
Yeah, so just a brief minute before lunch.